it's very easy to optimize the PK of a drug. And this is like basic, sometimes called hit to lead drug development. So hit to lead just means, you have a potential drug and you got to make it like a real drug. And it's not hard to elongate half-life. Like that is something that was just like extremely well understood in drug development. And if this was a real like drug discovery company, like at the very least you could do a controlled release. Controlled releases actually don't help you that much, but there are many, many, many well-known, like even undergraduate students would tell you what the ways are that could show you how to get this drug to work, with a longer half-life, to double or triple the half-life. And then there's also ways to get it to get into the brain more. When it comes to like an Alzheimer's drug, you don't want to take chances. And if your preclinical work shows that you're not getting into the brain very much, you want to get into the brain. This drug also has something called plasma binding. So they actually have relatively low plasma binding, which is good. It's around 33%. What's interesting about plasma binding is 33% in this case is bound to plasma and is not available for use by the body. Sometimes in science it's called free fraction. So their free fraction is sort of 70%. So 70% of the medicine can actually hit its target. Sometimes you develop a drug and the protein binding is like 99%. So we don't know what the bioavailability is. It is not 100%. Whenever you go through the stomach and the intestines, you will not have 100% bioavailability. In my model, I assume that they do have 100% bioavailability because I'm a nice guy. Reality, they could have as little as 50% or less. Who knows? We don't know if this is a substrate for one of the cytochrome P450 enzymes. We don't know if at all any of these things. So just because you're a small molecule does not mean you get good bioavailability. Ask ketamine. Ketamine cannot be given as a pill. It has like 5% bioavailability. And it's like a really small molecule. In fact, it kind of looks like 𝗕𝗏𝗏. So, if you're a very high affinity for some, cytochrome P450 enzyme, you very quickly could get metabolized into something that doesn't have the, say, lipophilicity to get into the brain. So there's a lot of issues with that. Now, not every company discloses the metabolic fate kind of chart of their drug, but we don't know a lot about this medicine, and some companies do publish that kind of stuff. So in any event, very little details here in medicinal chemistry campaign in terms of optimization, or we'd have a drug that is better than a 4-hour half-life. I mean, which is more likely? It's much more likely that they just winged it, put a fake drug into the clinic instead of actually doing a serious campaign. The biggest part of this is, and this is the part I really want you to think about, is how does this drug work if it's supposed to bind to that one protein? Now, I made an AlphaFold sort of X-ray crystal simulation, and it looks quite a lot like real X-ray crystals of filament A. Now, filament A is too big probably to make a crystal of, but they took partial crystals of it. And even just taking a crystal of the alleged 5-amino acid region that they think they bind to would have been nice, but I don't even think they even did that. But in any event, I did an AlphaFold, and you could do this too just by going to alphafold.com or whatever their website is. And you could see the 5 peptides that they say they bind to. And if you look at it, it's a very flat region. It's probably not even an active site of filament A. Filament A doesn't really even have active sites, but regardless, it's not a place of importance. So why would binding there help or hurt, even if you could bind there? I think there might be some small binding affinity to that region, but it's a flat region. It's not a pocket. In drug discovery, we generally want pockets that we can fill into. Think about, like a circular region, like a sphere that has an opening. You would want to fill that space. You'd want to fill it with a sphere-like kind of substance. And in drug discovery, it's not that different. It's called shape complementarity. And in essence, you do want to find a shape that fits into the space that you're filling. Sometimes called, you could call it space filling. And it has to be energetically kind of useful. In drug discovery, every bond we make contributes to the strength of the total bond or the total affinity. This drug doesn't have too many places to form bonds. It can form one really nice hydrogen bond with the carbonyl oxygen, but it can only form that bond one time, and it could do that to any amino acid, to be honest. So it's not really like specific, but you could get like 2 kcal per mole affinity that way. So none of this makes any sense, but we're going to go to the clinic anyway, just because. And we do a phase 1 study and we see a 4-hour half-life. If it were my drug company, I'd go back. I'd go back and say, hold on, guys, get me an 8-hour half-life or something. This is nonsense. Why would I move to phase 2? Right? Why do the phase 1 at all? You got a 4-hour half-life. If you have a bunch of these molecules, give me one with a 12-hour half-life. Throw them in some mice, throw them in some monkeys or dogs. Give me the one with the biggest half-life. It's because they don't know what they're doing. They don't have any people in real pharmaceuticals. They were studying OxyContin before this. They weren't actually doing real research, real drug discovery. There's nothing wrong with studying old drugs and making them work. Look at Jazz Pharmaceuticals, they did fantastic. But Jazz doesn't pretend they're scientists. Here, you're trying to make a novel drug. You can't do this with a 4-hour half-life. You need 8-hour, 12-hour bare minimum. If you somehow want the 4-hour, okay, get a controlled release company to help you at least. So you have all these issues. 4-hour half-life, what Vertex would do, Vertex put 3 different pain drugs, sodium channel inhibitors, into the clinic. They canceled. As soon as they got the results in phase 1, they say, nope, we're going back. We're going to try again. Nope, we're going back. We're going to try again. And their third one looked good. They moved into phase 2, phase 3. This is important in medicine. You have backups. Backups are critical. So they go into the clinic. The phase 1 has these terrible results. They said, oh, let's do phase 2 anyway. The FDA does not, by the way, care how much you develop the drug, as long as you aren't developing something toxic. You can call it phase 2, you can call it phase 3, you can call it phase 1,000,000. It doesn't make a difference. All they're really there to do is make sure that you're dosing a safe drug. After that, if you file for approval, that's when they come in and they care about your efficacy. But you can fail every trial you want, according to any objective or subjective measure you please. They don't stop you from doing a phase 2 or phase 3. As long as you have the tox coverage, which means you've studied in animals, how long you expect to study in humans. So if you have 6 months, if you're going to do a phase 2 with 6 months of human data, you got to do the 2 species rule is what it's called, 6 months of toxicology in 2 species, usually mice, rat and dog. So as long as you've done that, they don't care what you do from there on. Now, FDA approval is a different story. They're going to want to look at every piece of data 55 times over before they'll actually, give you approval. So they do the phase 2, and this is where the fraud kind of triples down. They tried to do a phase 2A that's 28 days long, presumably because they have 28-day tox data. And they do a word memory test, and the data comes out terrible. It actually looks like the placebo is better than the drug. That's not good news if this is your only medicine. And if you admit failure here, which is what you're supposed to do, because why spend $100 million on a drug that doesn't work? Or $200 million or more? Just admit you failed. Go to the rest of the drugs in your portfolio. Wait a second. They don't have any other drugs in their portfolio. This is all they've got. And admitting failure again? Maybe the board would fire me. Maybe it's better to just lie or fake it or just assume that, okay, maybe it does work. So what do? Dr. Burns takes the results and throws out patients. This is after getting the results and knowing it failed. Throws out data after data after data until what? Until it looks like the drug is better than the placebo. This is called lying. This is called fraud. This is a crime. And Dr. Burns had to step down because the SEC charged her with fraud, which is what she did. But nobody was told this. When the results came out, they said, look, we did the phase 2. It looks like the patients are doing better on drug than placebo. They're forgetting less. But she had a dirty lie. The dirty lie was, well, actually, they were doing worse. I changed the data so that it looks like they were doing better. Stock goes up, sell some stock, get some bonuses, keep paying a big salary. Now we're ready for phase 2B. Phase 2B is even worse. A lot of people don't understand this. And again, this is not, I'd ask yourself, Is this the first time you've ever been on a clinical trial? Did you understand most of what I said for the last hour or two? If the answer is no, the best thing you could do for yourself is whatever your position is, just take half that position. If you're good at this game of betting on clinical trials, let me tell you something, Cassava will not be your last victory. If you're actually good at telling whether or not a clinical trial can work, if that's a skill of yours, there will be 100 more Cassavas. And you will become one of the richest people in the world. There's a famous fund, I won't name who, but basically they kind of did this with very little capital, and they built it up. They built it up, and they actually did have the skill, and they built it up to billions of dollars. Actually, no, several of these. I'm thinking of one in particular, but you know, if you're good at predicting phase 3 outcomes, why is it only going to be for this drug? It won't. It'll be for other drugs too. You'll find other Cassavas, and you'll be one of the richest people ever. Because you'll make a 10x in Cassava. Yeah, maybe instead of having 100,000 on the line, you'll have 50,000 on the line. Fine. You listen to Martin Shkreli. And you have 50,000 on the line. But now you'll have 500,000. And you'll find the next Cassava, since you know how to find these, right? And next time you do this, you'll make 5x. And you'll have 2.5 million. And you're not going to feel worse just because you had half as much. But don't be a hero. Understand that you maybe not be God's gift to biotech stock picking, especially if this is your first shot. Invest half or less of what you think you have. And if you're right, hey, do it again next year. Do it again the year after. Do the math on how many Cassavas you need to become a billionaire. It's like 5 or 6. It's not that many, maybe 10. But if you know how to pick them like this, why go all in on this one? You might lose it all too. I think you will. So just do half or a quarter. And if I'm wrong and you're right, okay, next time go mega size. Get some confidence and some wins under your belt, right? Why would you go? You want to be a karate fighter. Why would you, or a UFC, fighter? You just got your first lesson. You say, I want to go to the ring, Brock Lesnar right now. Give me that guy. Who's the Irish guy? The Irish boxer, Irish UFC guy. Give me Conor McGregor. I want to fight him. Well, hold on, dude. You just learned how to fight. Maybe just fight the local gym guys first before you fight in the UFC. No, I want to put my whole portfolio on this. I've never done biotech before. What are you, crazy? You want to gamble? You just like to gamble. Well, that's fine. You want to gamble, gamble. I'll take the other side of the bet. But if you actually want to be good at this, you can become good at this. But see if you're going to be good at it before you imply that you're going to be good at it by just betting randomly huge amounts. So the phase 2 is even worse. And this is the biggest point of contention. Folks really don't understand this part, and it's unfortunate. In the phase 2, patients got, in the phase 2B, I guess, the controlled withdrawal study, patients got a year of open label 𝗠𝗏. Open label means they know what they're getting. The doctor knows what they're getting. There's no mystery. There's no control. So anything can kind of happen in that environment. And you can't really compare controlled results to uncontrolled results. It's like comparing a race where they had a timer and a race where they didn't. A timer, an official track, a referee, all that stuff, to a backyard race with nobody recorded and nobody's watching. They're really two different things. And for some reason, in the 2-year open label study, and there was this 6 months where it wasn't open label we'll talk about that are very, very important. But that 2-year open label study, patients got worse. They got sicker. Just as you would expect Alzheimer's patients would. But what the company's done, and people have been fleeced by, they're pulling the wool over people's eyes, is they said, the better half of those patients in this trial did better than the worst half. That doesn't sound very impressive, does it? That sounds like a tautology. But what they did is they called this half, they ordered the patients by how well they did, and they picked the top half. They said, wow, these guys did better than the bottom half. Well, yeah, of course, they're the top half, right? But they called them mild to moderate, or mild, I should say. And they called the other half moderate. And they showed the charts. And lo and behold, the mild did really well. And the moderate did really poorly. But that's because they ordered the patients and said, these patients had less disease and they did fine. They didn't rapidly decline in function, and these guys who had advanced disease rapidly declined in function. And if you average them, you take the total group, they declined in function, and they declined kind of on pace, in fact, exactly on pace, as to what you would expect them to decline. They did exactly as much. And I built a little database of Alzheimer's studies. And bulls will say that over the 2 years, that group, that mild group, did better. They stayed stable, didn't lose any function at all over 2 years, which is a big deal for Alzheimer's. That's never been seen before. Not only is that false, but this is an uncontrolled study. So over 2 years, the best half of these patients, which was kind of defined by the company and never published, by the way, and this is a company that's been proven to be fraudulent, so I'm not sure we can trust this press release regardless, but even if you trusted it, the patients that stayed flat over 2 years, not only have we seen this before, and there are numerous publications where we've seen this, but we don't know if they're even telling the truth at all. But this is just the better half of a data set where if you look at the average patient in this group, they're right in the middle of what you'd expect from these trials. And it's not a controlled trial. Uncontrolled trials have a lot of bias. So this data's, to me, actually proof that it doesn't work. And the most important part of this trial, which you can't form opinions on trials on uncontrolled data, the controlled data, they did a 6-month controlled experiment, which is what you're supposed to do, where after 1 year of open label, they did 6 months of control, and they did back to open label for 6 months, so a total 2 years. The control data failed. And this bothers people because when they hear fail, they know that's bad news. But when your p-value of your primary endpoint is less than 0.05, your trial has failed, period, the end. That's what a trial is. It's a pre-specified test. It's a statistical test, something like a t-test. And if you want to learn how to do these tests, I made a whole repo on my GitHub you can download. You can look at the source code, decide if it's fair, or you can go to graphpad.com or Google t-test calculator, and you do a t-test. And if the t-test fails, you basically have a failure in your hands. You know that the drug doesn't work. But the company knows that people don't understand this concept that intuitively. It's a pretty difficult concept to understand. They looked at the results and said, hold on a second. If we take these results and assume that whichever half of these patients did better against placebo, we'll just say that that's the half that we're going to go with. And in this case, it was the mild patients. And you can easily retrofit a new thesis. You can say, well, the mild patients, they're not yet so brain damaged that we could actually still help them. But what's funny is you can actually claim the opposite too. If the moderate patients did better, you could say, well, the mild patients didn't yet present with enough disease. I can't treat them if they're not sick yet. Their Alzheimer's is still so early. I need people who are really sick so that I can show a profound benefit because the sickness is significant. And if I stop that fast decline, I can show a better benefit. I've seen companies do both. I've even seen companies go from one to the other and back. You cannot simply look at your data post hoc and then decide why your post hoc observation is good for you. The only thing you can do in science is make a hypothesis and test it. And they did that, and it didn't work. The drug and the placebo had the same effect. They didn't even dare publish the p-value. They just said it was non-statistically significant. In these studies, the ADAS-Cog standard deviation is typically around 6 or 7. So they said they had a 0.5 point benefit versus a 6 or 7 point standard deviation. You don't need to be a great scientist or have done lots of experiments to know how spurious and irrelevant that is. But they said that in the mild patients, they did do better. But here's the thing. There are not too many diseases where drug doesn't work at all in moderate patients, but works really, really great in mild patients. I can barely think of examples. I was looking at Drug Bank last night. It's a great site from University of Alberta. It has every single drug that's ever been approved in any country. And I was looking for them, looking through it and going through every drug to see if there were drugs that work only in mild disease but not severe disease. And I would venture to say 95% of drugs do not work that way. There are 5% that do. And just as there are 95% of drugs are not protein-protein inhibitors, there are 5% that are. So you have to believe in Cassava, you have to believe that a post hoc observation is valid, which is extremely rare in medicine. That not only that, but that there's a difference between mild and moderate patients where a light switch flips off if you're moderate and it doesn't work. Imagine if your asthma inhaler, you have asthma and you took your asthma inhaler and it doesn't work. And you say, why doesn't it work? Oh, well, you have moderate asthma. But I'm just on the borderline of moderate asthma. Well, as soon as you go from asthma 21 to asthma 23, the inhaler stops working completely. It defies science. It defies things like the law of mass action, like basic scientific principles. It should still work. And people point to cancer. Look, Cassava lungs point to cancer. This is actually completely wrong. Most cancer drugs work, even if you're far along. They just don't give you much of a benefit. Chemo works late stage. Chemo works early stage. Chemo works adjuvant. Keytruda works late stage. Keytruda works early stage. Keytruda works adjuvant. There are really not that many drugs that behave according to this purported behavior. There's a handful, but even then, by the way, they still work in moderate. They just don't work as well. It'd be very weird for them to not to work at all. I would say only a few % of drugs work that way. So this must be that, right? And this must also be a PPI. Even though there's like 2 FDA-approved PPIs, this must be the third coming up, right? And this is a subgroup analysis that you can trust, of course. And this is a drug that was well designed, despite not having any chemists around it. And the guy who worked on it has been arrested. And the prior phase 2 data was never been published, not once, neither trial. And we know that some of it was completely doctored. It's very hard for all of these necessary conditions to be true, because none of them are individually necessary and sufficient, but you can all agree that all of these are necessary. If anybody has any questions, or if there's a bull here, I'd love to talk to a bull. So you have this phase 3 coming up, and I've looked at 6 recent Alzheimer's phase 3s, and the effect size from phase 2 always shrinks, always. Why? Well, law of large numbers. And it just sort of ends up being that you don't get better results in phase 3 than phase 2. Just hasn't happened. So to me, I don't think there's a chance this can work, because every one of the conditions I mentioned, the problems with the PK, the fraud in phase 2, the failure in phase 2B with the controlled withdrawal. Oh yes, this is the last necessary condition. The phase 2B withdrawal failed, but there's an explanation. Hold on. There's an explanation. The explanation is that the first year of drug that they've got, once half the patients got withdrawn to placebo, the other half got to stay on. But at the end of those 6 months, they didn't have any changes in outcome. Barely any difference. Either in mild or in moderate. But the mild one is a little bit better than the moderate, which had no effect. But it still didn't have really any effect that, in my eyes, that was statistically significant either. But the theory being advanced is that the drug is so good that it keeps working throughout the 6 months, even though you're not taking the drug. Now, I've heard a lot of copium in my life, and a lot of cope. But this is cope that is off the charts. When you stop taking a medicine, it stops working. Try it yourself if you have some prescription medicines. I don't think you will. And the reason for this is simple pharmacology. This is why we care about half-life. This is why we care about all of these things. Because there's never been a medicine in history that works that way. The sole exception is infectious disease, where, well, if you kill the infection, it's gone. Vaccines work this way, I guess, because you build up a permanent immune response. Even vaccines sometimes need boosters. There are very, very, very few drugs in my mind that I know of where you have a 6-month carry-on effect despite stop taking the drug. What normally happens is the day you stop taking the drug, you start to decline rapidly because you're not taking the drug anymore. It's no longer helping you, however way it's supposed to help, which, as we've mentioned in this case, there is no real answer to that. there's this idea of a misfolded protein. It's so absurd I won't even mention it. Filamin basically cannot be misfolded. It doesn't make any sense. The conditions that result in misfolding are usually pH conditions or mutations. Once the protein has docked and like formed and is there, it can't be folded or not, or like refolded or any of those things. There's a program if you want to try to learn it. I'd say 99% of you would not be able to operate it, including myself. But if you want to try a program called AMBER or GROMACS, you can do something called a molecular dynamics simulation. I actually used to program software like this. And you can actually watch the protein in 3D. And you can watch it wiggle around in nanoseconds. You can simulate like in a couple picoseconds or nanoseconds. I know the company likes to mention femto. But for a couple of nanoseconds, you could watch the protein wiggle about. Proteins are not living things, but they do respond to steric hindrance and molecular forces. And you can actually watch this protein wiggle about. Proteins are real living things, almost. They look like it. And with sophisticated software, you can simulate this. This protein is never misfolded, I would venture to say. Because if it was, you could actually, I don't know, take a photo of it with the X-ray crystallography. You could show that it forms aggregates. You could do things. And none of the science adds up here. So if a protein were misfolded, is this drug refolding it? Well, hold on a second. If it's refolding it, is it refolding it by binding to that peptide region that we're talking about, that solvent-facing 5 amino acid region? By binding to that region, we're refolding this whole 2,700 amino acid protein? Doesn't make sense. How could it be? If you know anything about protein folding, you know that you usually have to get it in the Golgi or the ER with different pH conditions. By the time it's in the cell, the pH ain't changing. If your cellular pH is changing, you got a bigger problem. You're going to be dead pretty fast. Or there's some chaperone or something that's going to be forcing this folding. Remember, folding happens on a Gibbs free energy gradient. Folding is simply the best conformation of a protein at that forms lowest energy. To go against that concentration, there has to be something forcing that. Usually it's pH, but it could be a heat shock protein or something like that. But why would this small molecule binding to this 5-peptide region magically refold this protein? And by the way, with a 4-hour half-life, how are we refolding all the filament in the brain all the time? It'll just fold back because whatever concentration gradient it was going against in the first place, that it needed this drug, right, to quote unquote refold. Once the drug is gone, it'll refold in the other direction. This is, basic entropy. So again, refolding at cellular pH doesn't really happen. Vertex's drug actually makes structural changes in the ER at low pH because that's where, if you're going to do any refolding, that's where you're going to do it. But that's because these CF patients have mutations. So they need refolding. There's never been anyone who's talked about filament A being folded incorrectly. And by the way, this is all being done in this isoelectric assay. This is not an assay for refolding. This is not how you measure refolding. So this is sort of an absurd way to try to come up with a new mechanism. Because the old mechanism, they know, doesn't make sense. Alpha-7, nothing to do with Alzheimer's. If it did, we'd make alpha-7 drugs all day long. Skip the filament. Who cares about the filament? I'll just make an alpha-7 drug. I'll knock out alpha-7. I'll delete alpha-7. Whatever it takes to save Alzheimer's. I know somebody in my family will get it someday. I also like money. I like making billions of dollars, so lots of other people do too. So we'll just make an alpha-7 drug. But that doesn't make sense because we've tried alpha-7. So instead, let's talk about refolding. But again, if you know anything about proteins, this doesn't really add up. Why would it refold incorrectly in Alzheimer's patients? In the cell, are the cells of Alzheimer's patients different from the cells of brain cells that don't have Alzheimer's? Why? Did the pH change? If the pH change is strong enough to cause protein denaturing, you'd have a lot of other problems. And I don't think 𝗕𝗠𝗠𝗡 is going to fix all of them. Can it refold every other protein that's broken? It just, the science isn't here. And the phase 2 data, again, this is being fooled by division. You can take any result, divide it by 2, and by definition, for example, if you threw 100 coins, the first 50 coin tosses and the last 50 coin tosses, one of those 2 groups did better. Is it because you tossed the coin better the first half or the second half, whichever one it might have been? No, it is not. It's because of chance. And if you take that group and say, hey, hold on, the first half I tossed 60% heads. The second half I just tossed 50-50 as normal. It must have been doing something that first half that contributed to my coin tossing. No, everything has to make sense. And you could invent some reason. Well, when I was tossing the first 50 coins, I was wearing my Lucky Yankees hat. Well, now you have to toss the coin 5,000 times. And your Lucky Yankees hat will not help you. Because when you're only tossing 80 coins or 100 coins, and this mirrors how many patients, for example, you might just get lucky. In fact, you kind of are bound to get lucky as the number of subgroups you take approaches infinity. The number of lucky chances you're going to have to explain the situation will also go to 1. There's a protein genotype called, or I'd just say a genetic genotype, ApoE, where if you have ApoE4, you have rapidly developing Alzheimer's. Scientists don't 100% understand why, but everyone agrees that you do not want ApoE4. You might even want to get yourself checked out to see if you have ApoE4. If you have ApoE3, you're normal. If you have ApoE2, you're actually protected. And everybody in this room right now listening has one of those 3. Which ones do you have? I'd rather have the ApoE2. Maybe you were unlucky and you've got ApoE4. Now you have an axe to grind. Now you have to care about Alzheimer's because this affects you. ApoE4, maybe some drugs will affect people who have ApoE4 more than ApoE2 or ApoE3. Well, there's more subgroups I can take, right? What if you're on acetylcholinesterase inhibitors? Maybe that's a subgroup. How about if you're on amyloid beta antibodies? Using all these subgroups, I can keep defining new groups. And with enough groups, I can show you a subgroup that did really, really well. The problem is it's not a priori. I didn't pre-specify. I didn't say, I'm going to show you that mild patients are going to do better than moderate. I decided that after I saw the data, not before. It's a huge difference. And if you know anything about medicine, and if you've done this for a living, if you've bet on clinical trial results for a living, you know that these subgroups never replicate because we see them after the fact. They're not predicted before the fact. And it sounds like, what's the big deal? If they did better, they did better, right? No. There's a statistician called Bonferroni who looked at this a long time ago, and he came up with this principle of multiple comparisons. You actually have to adjust the p-value in those cases. You have to actually make it exponentially harder. Because why? Because you have the data. You already can see the data, so you're tempted to contrive the answer. Richard Pazdur, head of FDA's oncology division, once famously said that looking at a post hoc subgroup is like shooting an arrow and then painting the target after you shoot it. This is just not how it works. You have to say, I'm going to hit that bullseye. You take the bow, pull back the arrow, and you hit the bullseye. That is how science works. You don't say, well, you know, I hit the wall. I think I hit a bullseye. Let me draw the bullseye chart. Yeah. Turns out I hit the bullseye. How about that? You know, it really is disingenuous. You're smarter than that. It's too stupid to think this way. Don't be fooled by this. It never pans out. Again, I don't care if you lose money or not. I kind of need people to lose money if I'm going to make any. So I don't really feel bad. But I feel like, again, it's just my narcissism that I'm willing to talk about this. And since I started live streaming and since I started talking about biotech publicly, it's been a long time. I've been doing this for a really long time. And I always get the same response from people who are long. And it's usually their first investment in this field, and they're certain that they're right. And I just sort of shake my head and laugh, and, you know, that's fine. You're right, I guess. We'll see. But I feel like this has been a really interesting case, and there'll be cases like this again and again for the rest of forever. As Groucho Marx once said, a sucker is born every minute. And